And with that, I'll pass it over to Katarina, in order to introduce our first speaker.
Great, thank you so much, Kelsey. Okay, so our first speaker is Katarina Klett. Katarina, as an undergraduate student, quickly recognized her passion for solving clinical challenges in the lab. After earning her bachelor's degree in chemical engineering from the University of Pittsburgh, she moved across the country to continue chasing that dream. Katarina is currently a stem cell biology and regenerative medicine PhD student at Stanford University, where she is leveraging biomaterials and intestinal organoids to better understand how our intestines function in the Heilshorn lab. She was awarded the National Science Foundation Graduate Research Fellowship in 2020. And with that, I'll just pass it over to Katarina.
Good afternoon, everyone. Just give me a second as I navigate the screen sharing here. So far, you should see a picture of my puppy, maybe. You see that? Wonderful. And now do you see my slides? Okay, wonderful. Thank you so much. Let me just begin by saying, number one, thank you to Katarina and to Kelsey for inviting me to speak about my work here today. It is such an honor to be able to chat with you all, and I'm really excited to share some of this recent work that I've done. So for those of you who I haven't had the pleasure of meeting yet, my name is Katarina, and I'm currently a PhD student at Stanford under the mentorship of Sarah Heilshorn. And today I'm going to share a story about how we developed a tunable engineered matrix for the culture of human patient-derived intestinal organoids. And so before I spend the next 20 minutes or so talking about intestinal organoid culture, I want to make sure that we're all on the same page, first and foremost, about what intestinal organoids actually are. And so intestinal organoids are three-dimensional cell structures derived from either human or animal biopsies. And our intestines, and so intestinal organoids have three core criteria. The first is that they mimic the native architecture of our intestines. And our intestines is organized into these things called crypts and villi, which are shown here in this schematic. What you'll notice is that the crypt contains highly proliferative cell types of the intestines, including the stem cell niche. And as these cells differentiate and mature, they work their way up the villus until they eventually die and are sloughed off into our intestinal tract. Organoids also contain many of the cells that we see in our own native intestines, like Paneth cells and intestinal stem cells. And finally, they typically have some sort of functionality, like maintaining barrier integrity. And so the current standard for growing intestinal organoids today is in Engelbreth-Holm swarm matrices, also known as EHS matrices. And this image here on the right is taken from a well-known Sato Cleaver's paper where they first demonstrated that they could form an entire intestinal organoid from a single LGR5 positive stem cell. And this was done in an EHS matrix. And so you might be thinking, wow, it seems like EHS matrices are really effective for growing intestinal organoids, and you would be totally right. That's why we use them as the current standard. But for scientists like myself, who is interested in probing questions surrounding how the extracellular matrix impacts intestinal biology, there are several key limitations that make EHS matrices maybe not the best option for us. The first is that EHS matrices are derived from mouse sarcoma. And so that means they contain hundreds of different proteins. And so you might wonder why does that matter? But if I wanted to tease out specifically how maybe one or two proteins are impacting the cellular biology of my intestinal organoids, it would be challenging for me to do that with so many other proteins present. The next is there's low batch-to-batch similarity between EHS matrices, which means that sometimes reproducibility can be a challenge. And finally, for someone who might be interested in how tissue mechanics impact cellular biology, like stiffness or stress relaxation rate, there's limited tunability in EHS matrices. And so I've probably already beat myself to the punchline here with the title of my talk, but our lab has developed an alternative matrix that overcomes all of these limitations. And we title this matrix the HELP matrix. And the HELP matrix has two key parts. The first is what we call an elastin-like protein, or ELP. And ELP has two pieces that I'd like to highlight here today. The first is a fibronectin binding motif known as RGD. And RGD mimics the cell fibronectin ECM interactions that we have in our own extracellular matrices. This RGD can also be swapped out with an RDG domain that is non-binding, so unable to interact with cells. ELP also has something called an elastin-like domain that we can modify chemically in-house to have a hydrazine group. And this will become important in a second when I describe the H portion of the help matrix. And so you might be asking yourselves, why is someone like me who wants to grow intestinal tissue so interested in this fibronectin binding motif? Well, it turns out if we look at the structure of our intestinal tissue, that there is a high concentration of fibronectin in our intestinal crypts. And I remind you that this is the area that has high proliferation in the intestines. And so you can imagine if I want to develop a matrix in which we can initially form and grow a number of intestinal spheroids or organoids, it would be helpful to mimic a similar environment. And so the next part of the help matrix is hyaluronic acid, also known as HA. And we can modify HA in-house to have either a benzaldehyde or an aldehyde group. Now remember, I told you that hydrazine group was going to be important. So the hydrazine is what bonds to either the benzaldehyde or aldehyde groups, and those are the bonds that are holding together our gels. And so again, why is this so important when we think about growing intestinal tissue within a matrix that has something like hyaluronic acid? Well, it turns out previous work had demonstrated that if you administer exogenous HA into the mouse, it increases the number of LGR5 positive stem cells in the intestinal crypt. And so if you look at this plot with me, here on the left, we have a control that was untreated. On the far right, you have a PEP1 negative control, which is an inhibitor that blocks HA from binding to its receptors. And when we compare that to the group that has hyaluronic acid, there's a significant increase in the number of LGR5 positive stem cells. And so again, if we initially want to create a matrix that supports this formation and growth, it could be really advantageous for us to have hyaluronic acid within the material. And so the first thing that we were able to demonstrate is actually forming these intestinal spheroids from single human primary cells in our material. And so just to provide some historical context with what had been done in the field predominantly, were that spheroids were grown in EHS matrices. Those whole spheroids were then removed from those matrices and then re-embedded into engineered materials. And we call this re-embedding. But what had been really a challenge in the field up until recently was forming intestinal spheroids from single human primary cells. And we call this from a dissociated state. And so if you'll follow me over here to the right of this slide, the first two columns are spheroids that have been grown from a dissociated state, so from single cells. And the two columns on the right are spheroids that have been re-embedded. So again, that was initially formed in an EHS matrix and then removed from the EHS matrix and re-embedded within one of our matrices. And so in this top row, what you're seeing is an ELP only gel. And what you'll notice is that we were unable to form intestinal spheroids from single cells in this gel. We also noticed that when we formed these intestinal spheroids in EHS and then re-embedded them in this ELP only matrix, they didn't maintain the polarity that we were expecting to see. Now, what do I mean by polarity? Well, it turns out that our intestines are actually a polarized tissue. What does this mean? So if you look at these intestinal spheroids that I have pictured here, you'll notice that there's a ZO1 apical in border in all of them highlighted in a green stain, and a beta-catenin basal out border highlighted in red. And so we were very excited to be able to grow spheroids from single cells, both in HELP and of course EHS, but also be able to maintain this polarity following re-embedding. And so if we look at this quantitatively here in the bottom left, what you'll notice is what we see by eye as well. And that is that there's a significant increase in the ability for spheroids to form within EHS and HELP matrices compared to ELP only gels. And so you can imagine if we want to grow and maintain these cellular cultures for long periods of time, it would be really beneficial to be able to passage them within our gels. And so we've demonstrated that we can passage them for over 15 passages. And here we have two images demonstrating what they look like across passages in our gel. So on the leftmost picture, this is in the first passage in HELP, and then passage 5 in HELP here on the right. And if you follow me over here to the right of the slide where we have this plot, we were very, very excited to be able to demonstrate that if you passage spheroids within HELP for an equivalent number of passages to those grown in EHS matrices, here we show passage 12, we could grow spheroids that are statistically the same size as those grown in EHS matrices. Now, why is this so important? This means that we're growing spheroids that people are growing at the current state-of-the-art size. And so that was a really exciting finding for us. And so, of course, we were thrilled to be able to grow intestinal spheroids, but we want to actually be able to differentiate them into organoids, because I told you at the beginning, that's the powerful model that we're really looking to recapitulate. And so here, I remind you, we start from single human primary cells, and we seed them within our HELP gels. Within 1 to 3 days, we form an early spheroid. Early spheroid has already formed a lumen, and you'll notice that the nuclei are aligned parallel to this lumen structure. If we then wait 7 to 9 days, you'll notice that the spheroid is starting to polarize. The epithelium is starting to thicken, indicated by this thicker actin border here in magenta. And you'll also notice that the nuclei are now starting to flip perpendicular to this lumen. Finally, if we differentiate our spheroid for 5 days using differentiation media, you'll notice that the organoids form these undulating lumen structures, which has been previously shown to be indicative of differentiated human intestinal organoids. And so let's zoom in a little bit more. We wanted to see specifically what intestinal cell types do we have in these organoids. And so just to orient you, the top row here are organoids that were grown in EHS matrices. The bottom row are organoids that were grown in HELP. And the far column here on the left is a mucin 2 stain in white. And mucin 2 stains for goblet cells. And goblet cells are a really important cell in the intestines as they secrete mucin and make up the mucin lining of our intestines. The center column here is a chromogranin A stain in yellow, which is a known marker for enteroendocrine cells. And enteroendocrine cells are important in secreting hormones within our intestines. And finally, here on the far right, you'll notice a lysozyme stain in green, which is a marker for Paneth cells. And Paneth cells have a number of important functions within the intestines, but I would say one of their maybe most important is their ability to maintain the stem cell niche. And so, of course, I love looking at organoids in 2 dimensions, but because I had the opportunity to be able to give this talk today, I wanted to be able to show a video as well, because I think it helps to better visualize what this looks like in the 3 dimensions. And specifically here, we're going to be going through an organoid Z stack. And I want you to pay attention to this actin stain in white, where you're going to notice these undulating lumens that I mentioned. You're also going to notice a couple goblet cells highlighted in the mucin 2 green stain. So here's some goblet cells in the bottom, those undulating lumens I mentioned, a goblet cell in the top left, and we're just working our way through the organoid. So something that may have caught your eye in my previous slides is that we were unable to grow intestinal spheroids within ELP-only matrices. Well, it turns out that previous work had demonstrated that stiffness plays an important role in spheroid formation. Recognizing this crucial role, we stiffness matched an ELP-only gel to HELP at a stiffness of around 1 kilopascal. This is shown here in this plot on the left. We then seeded these single intestinal cells within the HELP and ELP-only matrices. We saw a stark contrast in the ability of each matrix to support spheroid formation, which I think is pretty clear between these 2 images. We also noticed that we saw the expected polarity within the spheroids grown in the HELP matrix, whereas we didn't see that polarity at all in the ELP-PEG matrices. And this matched what we saw quantitatively here on the right, which was a significant increase in the formation efficiency of organoids grown in the HELP matrix compared to those grown in the ELP-PEG gel. This led us to hypothesize that HA must be playing a key role in the spheroid formation we observed in our HELP matrix. And so a question that you might be asking yourselves, which is certainly one that we were asking, is how does hyaluronic support intestinal spheroid formation and growth in the HELP matrix? So HA is known to bind a receptor known as CD44, and CD44 has been shown previously to be important in cell migration and proliferation. And so we thought that this might be a receptor that's important in driving this initial formation and growth that we saw. And so diving into this a little bit further, we did flow cytometry experiments to see the percentage of cells that were CD44 positive. And so these were spheroids that were grown in either the HELP matrix or EHS matrix, HELP here in purple, EHS in black. And what we saw is that cells that were grown in these matrices across gels had high percentages of CD44 positive cells. Interestingly, we noticed two key points when we repeated this experiment. The first was that there wasn't a statistical difference between the percentage of CD44 positive cells across both gels. In other words, CD44 was always high regardless of the gel. But what we did notice, or another thing that we noticed, was that when we looked at variance, spheroids that were grown in the EHS matrix had a significantly higher variance than those grown in the HELP matrix. And again, I think we believe that this is primarily driven by that batch-to-batch variability that I mentioned at the beginning of this talk that's typically seen in EHS matrices. Okay, so I hope by now that I've demonstrated that we can robustly form and grow intestinal organoids within our HELP matrix. Now the question is, how can we actually tune this HELP matrix to better understand how matrix properties might impact intestinal biology? So first we can tune the RGD concentration. And I remind you that RGD is the fibronectin binding motif that allows us to mimic those cell fibronectin interactions that we see in our own extracellular matrices. And so we can tune RGD in our material by doping in the scrambled RDG domain. Thus, by adjusting the amount of RGD seen here in dark pink on the left, or the amount of RDG seen here in the center in light pink, we can specifically tune the concentration of RGD in our material. Next, we can modulate matrix stiffness by changing how modified our HA chains are. For instance, if we highly modify our hyaluronic acid chains with benzaldehyde groups, like the matrix seen here on the left, there will be a greater number of bonds that can form between these benzaldehyde groups on hyaluronic acid and the hydrazine groups on ELP. This results in a stiffer matrix. However, if we modify our hyaluronic acid chains at a lower percentage, like the matrix shown here on the right, fewer bonds can form between these pink benzaldehyde groups and these hydrazine groups highlighted in orange. And this results in a softer matrix. And so if you'll follow me here down in the middle left, you'll notice that when we look at the stiffness of these materials, our stiff elastic matrix is around 1 kilopascal, whereas our softer matrix is a little under 500 pascals. Now, although we are changing stiffness, we can also maintain viscoelasticity in our material. So for those of us that maybe aren't as familiar with the term viscoelasticity, viscoelastic materials are materials that exhibit both viscous properties like a fluid and elastic properties like rubber. And it actually turns out that many of our organs are viscoelastic because of their high water content. One way to demonstrate this is by pushing the skin in on your arm, and you'll notice that it quickly bounces back. This is because your skin is dissipating and relieving the stress that you just applied. Our extracellular matrices undergo a similar molecular level remodeling after being deformed by cellular forces. In this case, you'll notice here in purple and in pink, both of these materials are slowly stress relaxing with stress decay half times of around 18 hours. So what actually happens when we grow intestinal spheroids within these matrices? So it turns out that intestinal spheroids grow significantly larger and stiffer matrices. And just to orient you, the stiffer matrices are here on the left in purple with varying RGD concentrations. And then the softer matrix is here on the right in pink. We also notice that our spheroids tend to prefer matrices with higher concentrations of RGD, specifically between 0.5 and 0.75 millimolar. And so finally, we can modulate viscoelasticity within our gels. And we do this by tuning the amount of aldehyde groups in our gel here in blue. And so if we increase the number of aldehyde groups in our gel, it results in a faster stress relaxing material or more viscoelastic, like the material we see here on the left. However, if we increase the number of benzaldehyde groups in our material, seen here in purple, we can make this a slower stress relaxing material or less viscoelastic. Now, if you follow me here to the right, you'll notice that even though we are varying viscoelastic properties between these two matrices, we're maintaining a stiffness of around 1 kilopascal. Now here on the bottom right, you'll notice that the stress relaxation across these materials has a large difference, with the stiff elastic material here in purple having a stress decay half-time of around 18 hours, whereas the stiff viscoelastic material here in this blue has a stress decay half-time of around 30 minutes. And so again, what happens when we actually grow our spheroids in these materials? So we found a very interesting result here. And that was that if we grew the spheroids in this stiff viscoelastic matrix, we noticed that they were more sensitive to variations in RGD concentration. And again, to orient you, the stiff matrix is here on the left in purple, and the viscoelastic matrix is here in blue on the right. So we are very curious about this finding, and we're thinking about further exploring it moving forward. And so what are the key takeaways that I hope you leave my talk with today? The first is that our HELP matrix supports the formation and growth of intestinal spheroids from single human primary cells. Next is that these spheroids can be differentiated into organoids that contain the canonical intestinal cell types. And finally, we can tune this HELP matrix to help us better understand how matrix properties impact cellular biology. And so finally, of course, I want to thank everyone who has invested an incredible amount of work, energy, love into this project, specifically my mentor, Sarah, and my partner, Dan, who recently graduated with his thesis focused on this project, the incredible Heilshorn Lab, who is just a wonderful team to work with, all of the funding that helps me be able to do this work. And finally, I did put my email here because I know it can be a little intimidating to ask questions in a setting like this. It's awkward over virtual format. So if anybody would want to chat one-on-one, please don't hesitate to reach out. I'm always happy to connect.